So the background to these first few slides, and yesterday you might have seen, again, if you subscribe to our YouTube channel, we produce videos. We feel like they're a really good way to interact with people. The visual, the music, they kind of immerse you in a concept. And as we know, video is very, very popular in modern culture. So yesterday we put out a video trying to explain about SMAD4, and Armel and I, over the last kind of couple of weeks, and if you've been part of these groups for a while now, you'll remember at the beginning we talked about trying to find an analogy or a way to describe SMAD that just wasn't particularly as technical, but had relevance to something that we could just place in our minds that was perhaps a bit more, I wouldn't say normal, but, you know, a bit more relatable. That's the word I'm looking for. So Armel and I were working on this, and we came across this analogy of like a factory and a factory manager, and how SMAD acts as this factory manager and the processes that it goes through. So we produced this analogy, and really, again, this is a point where we would love your feedback on if these points make sense in this and concept. But Armel, I'm not going to do it justice enough. So you talk through the actual concepts of how we have put this analogy together, and then let's just really open it to the floor and go through the bits that people may or may not want clarity on, etc.
So the way we thought to explain SMAD4 was that SMAD4 is a factory manager. So it's going to control what's produced in the cell. So where to control would be the nucleus, and that's Kate found this analogy, that the nucleus would be the control machine, the control room, and the SMAD4 would go in the control room and tell the cell, produce that gene, don't produce the other gene. So the SMAD4 comes also from a gene, because every protein comes from a gene. So the SMAD4 gene produces the SMAD4 protein. And this protein tells the cells what to do, what to produce, and then triggers the function that are going to be used by those new genes that are on. So we can say that the SMAD4 is a gene switcher. It's going to say, okay, produce that, don't produce that gene. So in biology, those gene switchers, they are named transcription factors. So that's the role of the protein SMAD4, is a transcription factor, which means that SMAD4 is in the cell, is going to take other proteins, let's call them co-workers, and SMAD4 is going to hold their hands and bring them to the control room and bring them to switch on and off other genes, so to produce new proteins. SMAD4 can be named as a factory manager, and it receives orders from outside of the cells. There are other, let's say, messengers that are going to receive this signal from outside of the cell, and then we'll tell SMAD4, okay, now be activated, go get your coworkers, go to the control room, go to the nucleus, and switch on some genes. Sometimes SMAD4 can also not go to the nucleus, but activate other coworkers that are going to go in another messenger and trigger another function in the cell. But mainly its SMAD4 role is to take other coworkers, activate them, and bring them to the control room, to the nucleus, to switch on genes. So I hope that's tell me, just tell me your reaction. Do you understand what the analogy means, or do you have any? Is it clear? Do you have any questions?
I mean, I think it's a great analogy. I think it's laid out pretty well. And I watched the video. I think the video helps too. I did have a quick question. If you will go back, just relating to SMAD4 gene versus SMAD4 protein.
Mm-hmm.
And I'm trying to remember. But in the literature, I think if it's italicized, it means one thing, and if it's non-italicized, it means another.
Oh, yeah. We should have, yeah. So you have capital letters for the, I'm going to say something wrong. There is one capital letters for the gene or the proteins, and then small letters for the gene or the proteins. And I never pay attention to that. I think the capital letters are for the gene and the small letters for the protein. I need to check that. I'm sorry. That's details.
No worries. I
But
can
that's
look it up
true.
too.
That when you talk about to make the difference between the gene and the product, is in capital and the other one in small letters. We should have made this distinction. We can still modify, I think, the things. Good point, Don. So going in that direction about the difference between the gene and the protein, that's something I would really like you to understand. The difference between the gene, the DNA, and how can you go up to the protein, which is the real worker, in fact. So the proteins like SMAD4 are made up of building blocks that are called amino acids. And if you have a mutation in the gene, so it's the instruction of how to make a protein. If you have a mistake in the instruction, you will have a mistake in the building blocks. And I'm going to explain that more in detail afterwards. So the problem with Myhre syndrome is that you have some mistakes in the gene that are going to modify the protein in such a way that it's going to be too much active. So it's what we call a gain of function. So the analogy that Kate brilliantly found is that a gain of function mutation is likely giving the factory, if we stay in the analogy, the factory manager an extra tool or ability that they didn't have originally. And instead of really helping the cell to function properly, these new abilities are going to cause problems. So she's going on, and I think your analogy, Katie, is really brilliant. Imagine the factory manager now has a new gadget that turns on machine randomly or at the wrong time. This gadget, so what we call the gain of function mutation, it comes from the mutation, this gain of function. It makes the manager activate the production line incorrectly. So you have an overproduction of some genes, so it's what we call products in this analogy, or you block the production of some genes, so you underproduce some other proteins. So in the cell, a gain of function mutation in SMAD4 can cause it to turn on genes that shouldn't be, that should be off, or that should be on, but a shorter time, so you produce less, or it can activate processes that shouldn't be activated. Questions? No? Okay, great. So just now, I want to remind you that there is something very important I talked about last time also is for you to give blood samples and urine samples if you can for the patients, because we are gathering every year. We want to gather enough patients and the same patient over years, over at least 3 years, to be able to identify in the patient blood some proteins or other markers, other molecules that can show the stage of the disease or just that there is the disease. So that when we have potential drug, we can give it to the patient in clinical trials, look at that marker and say it's going, this marker is going back to normal levels, which means that the drug works, or the marker doesn't move, which means that the drug does not work. So that's a very, very important tool. And for that, you need to give your bloods every year, and we need as many samples as we can so that the statistics can be significant. If we look at 10 patients, then it's way more difficult to make statistics on the level of the different molecules than if we have 20 or 30 patients. So you can go to roadshows from Combined Brain. So we go through Combined Brain to gather those samples, and there are different other organizations that are having their patient conference or their convening, and you can go there to the other conferences and still give your blood for Myhre syndrome. So I really encourage you to participate to that. Also remember that you need to update your registry data every year. For the same reason, if we want to really assess new drugs, we need to know the evolution of the disease along the different years, along time, how the patients are progressing. And for that, you need to put your data in the registry. So don't forget to go every year in your registry and update your registry. So I would like you, really the purpose of those office hours, Q&A, whatever you want to call them, is for you to understand what we do at the foundation and to make you more knowledgeable about your disease and make you have a better discussion and communication with your doctors or the researchers, and really engage you in everything the foundation does. Have your feedback, and really maybe bring you by your hands through what we do, and have maybe your comments, know about what's important to you, what you don't understand. So that's really the goal of those meetings, is really for you to understand, but for me to understand what you think and what you feel. So please just interact with me, interrupt. It's a conversation more. I don't want it to be a webinar. I want it to be really a conversation. So for today, I'm going to start talking to you about understanding our toolbox and our portfolio. Our goal is to advance treatments. And for that, we have a strategy. And the first step of those strategy is to build a toolbox so that our portfolio of discovering treatment can be sustained. So what we work on now is creating the tools to test the potential treatments in cell and animals. So that's what we call the preclinical toolbox. And we work also on creating tools to test potential treatments in patients. So that's what we call the clinical readiness. Having those tools will allow us to accelerate the discovering treatments. We have 4 ways now that we focus on to discover new treatments. It's gene editing, cell therapy against fibrosis, and small molecules. So small molecules that the traditional type of drugs, and peptides. So I'm going to explain you a little bit more why you have that disease, so that you can understand why we choose to focus on those kind of treatments. So that's for you to understand what we do, what I call the MSF portfolio, on what we work. You need to understand why you have the disease. So I'm going back to 3 months ago, when I started to explain you the basic of biology, which is you have cells, you are made of cells. In the cells, you have a kind of other area that is delimited. It's like a bag where you have, that is called the nucleus, where you have your chromosomes, and in the chromosomes, you have the DNA and the DNA has genes in it. So part of the DNA is for regulation and parts of the DNA are genes. So the genes, they are the instruction to build proteins. So it's a code and it's really letters and words to tell the cells, now you are going to build that protein. And to communicate and give this instruction, we have only 4 letters that are A, C, T, and G. That stands for adenine, cytosine, I don't know how to pronounce it, cytosine, thymine, and guanine. So those are not just name, they are really molecules. So that's the molecule of adenine. You have here the T molecule, you have here the C molecule, and the guanine molecule. So when researchers are looking at a sequence or an instruction, they are not going to write all those different molecules. So instead, they write the letters, just the letters. So the sequence of nucleotides makes a gene, and it's written with the abbreviation letters A, T, G, C, because it would be impossible to make the whole molecules one after another. You couldn't read that. So that's the way we write a gene. What do we do with a gene? So you have all this sequence. Whoops. You have all this sequence here. And this sequence is going to get out. I'm going to go back to the other one. So it gets out of the nucleus, and something is going to read this code, all these letters, and make a protein. So how do we do that? So there is a ribosome, you don't need to know the name, that is going to read all these letters, and that is going to say, okay, when I see G, U, U, it means that I'm going to take an amino acid, which is a building block of the protein. I'm going to take that amino acid and link it to the chain. So this reader goes on the gene and reads this A, G, C, T, A, G, B, G, T, A, C. And every 3 letters, it stops and says, okay, those 3 letters is the code for an amino acid. If I can read G, U, U, it means that I have to put a valine. If I can read C, G, U, it means that I have to put an arginine. But imagine that there is an error here, a mistake in the DNA. And instead of reading C, G, U, sorry, I'm going to replace the C by, well, it's complicated because now it's a U, a C by a T, and then it's going to be UGU, which means that instead of putting an arginine, it's going to put a cysteine. And in fact, that's exactly what's happening in the SMAD4 gene for Myhre syndrome patient, is that at the coding sequence, the nucleotide number 1486, instead of having a C, so a cytidine, you will have a T. And if you look back in the coding, the genetic code, if you replace that C by a T, which is here, I know it's confusing, it's a U because you have an mRNA and not the DNA. So I skipped a step for you to be easier. So imagine that a U would be a T. So if you replace a C by a T, then you don't have an arginine, but you have a cysteine. And that's exactly what's happening here in Myhre syndrome. So what does it mean? It's just one amino acid. That should be nothing. So I'm going to bring you to a site, a website, where you can have really the SMAD4 gene in all the details. And I'm going to show you. So that's the chromosome 18. That's the place where there is the SMAD4 gene. And in this very small place, you have a mutation. And so when this DNA is transformed on mRNA, then transformed on proteins, what's the results? Oops, sorry. What's the results at the end? I don't know if you can see that really properly. So the difference will be, so we said it's 496. Where's the 96? So you should have an arginine. And instead of an arginine, sorry, it's where's the arginine? It should be the big one. Where's the arginine? Okay. So the arginine is not this one. It's not this one. Can we have a bigger? No. Oop, with 92, 93, 96. Okay, so you see, can you see the pink amino acid?
Yeah.
Okay, so you see this arginine, that's the normal amino acid that should be in the SMAD4. So that's the SMAD4, that's the protein. That's the way it looks. And instead of having this big, long chain of amino acids, the arginine, we have instead a very small one, which is a cysteine. So you can imagine that instead of having this very big arginine that makes bonds with the others, that hold hands with other amino acids in the protein, we don't have this one. We have a small one, like, I don't know if there is a cysteine somewhere that I can show you that it's really smaller. Well, anyway, the arginine has a long arm that can hold hands with another amino acid and bring the protein to be tighter. Instead of that, Myhre patients that have the mutation at the 496, instead of having this long arm that is going to hold hands and bring the structure together, they have a very small cysteine that is short and cannot hold hands with the other around. So it modifies the shape of the SMAD4. So if the shape is modified, the function is going to be modified. For the 500s, I think that's it. That's the isoleucine at the number 500s of the amino acid chain. Instead of having isoleucine, there is a valine. I think there is one valine here somewhere. Where did I see it? That's a valine. So you see the shape of a valine is totally different from the shape of an isoleucine. So the same, all the interaction that you would see, you see that it holds hands with this one, with this one. You cannot have that when you have a valine instead of this isoleucine. So the structure of the protein is going to be modified, then the function is going to be modified. Do you have any questions? We are getting really in deep biology and science here.
I mean, from my side, it's just fascinating to see a visual of it, but then B, just the big impact that has in the entire body. It's just, yes, it's fascinating.
Is there anyone that really don't follow and want to have more information? It's a difficult concept, so really don't hesitate. Ask questions. Well, I don't want to force questions. So, okay, maybe at the end, maybe when you, so where's the presentation?
I'm with Kate. I think it's really interesting, and it helps explain some of the previous conversations we've had about how the SMAD4 protein interacts with some of its neighbors like SMAD2 and 3. So seeing it spatially is really helpful.
Yeah, and I guess like back to the analogy of the co-workers and stuff, It's like they're getting their team together, but that team have not got the right instructions to make the product that's correct. So they're doing their best, but in that team, there is an error. And instead of making a red car or whatever it is that they're making, or t-shirts or bottles, there's a mistake. And it's, you know, but it has a knock-on effect. That team, it's affecting the entire team. And that's the thing I massively took away, Amel, from our discussions, is that thinking about SMAD4 and how we find a solution for this, it's not in isolation, because you can't just, the factory manager can't be the only one who's bought into the changes. The team are affected by any potential change. And so I hadn't realized before how the interplay with the other co-workers, the SMADs, is so, so important.
Yeah, exactly.
I was going to say, can you hear me?
Yeah.
Yep.
Okay, perfect. I can't tell if we're muted or not muted. I was going to chime in on what Kate and the other person was saying as well. And I think that, like, in perspective, like, I mean, being somebody who has it, like, when you're talking about, like, all the things like being impacted or whatever, and when one of those things is off and you, excuse me, then everything else is impacted as well. And you think about, like, our bodies, like you, I mean, literally, like, I know for myself that, like, if, like, you know, like, I'm having an issue with, like, my eyes or something, it usually manifests, and maybe we fix that, the eye issue, but then, like, something else gets you know, something else is impacted, and you fix that issue and something else is impacted, and it's been like that my entire life. And that's what I think that's what everything that you're speaking to really resonates with me, if that makes sense.
Totally. So I hope you understand that because of the mutation in the SMAD4 gene, then your protein SMAD4 has a different shape and does not properly interact with other proteins in all your body, not only in your eyes, not only in your T cells from your immune system, not only in your fibroblasts, but everywhere in your body. The SMAD4 protein that you produce from the gene is going to have the wrong shape, so it's going to interact not the right way with all the others protein.
Mm-hmm.
So let's go now to explain. So we try to make this analogy that SMAD4 takes the coworker SMAD2, 3, or SMADs 1, 4, 1, 5, or 8, and holds their hands and bring them on the other genes and tells the cells, okay, now you produce that gene or you don't produce that gene. So turn on and off. So to explain you visually, when you have a gene, so that's one gene, or I had animation, but if I don't do the slideshow, there is no animation. Okay. On one gene, you have the first part is called a promoter. So that's where the protein's transcription factor, so the role of SMAD4, is going to bind here and say, turn on or turn off the gene. Then you have a coding region, and that's where the code to make the protein will be. And then you have a termination sequence. So the role of SMAD4 is really to bring the coworkers on the promoter and say, okay, you turn on that gene and you are going to produce that gene so that the cell can make the protein corresponding to that gene. Oh, I don't have my animation. I'm so sad. So the approach we chose, yeah.
If you hit play on the right, will that not play? We can see the word play, but will it not do it?
Oh, yeah. Oh, thank you. So I was explaining everything, then I can do it again and have my toy and say, so the way we, the way it's going to be produced, that gene, is because, yeah, SMAD4 is going to bring its coworker SMAD2, 3 on the promoter of the gene and tell the cell, okay, now produce that gene. So the way we chose to tackle that disease, in fact, is first we said, okay, it's just everything in the whole body of Myhre patients starts because there is this mistake in the gene. So let's correct that mistake in the genes everywhere in the body so that we are going to have a SMAD4 protein that is going to work properly everywhere in the body. So that's one aspect that we chose to do, and we are working on gene editing with collaborations, of course. The next one, I told you that you have genes and that the role of SMAD4 and SMAD3 is to, SMAD4 is to bring the coworkers on the promoter. And the issue with SMAD4, because it has a different shape in Myhre syndrome patient, is that it holds the hands of the coworker SMAD2/3 too long. So it's going to tell the cells to produce those genes way too much. So what we thought to do is to have something that will separate the coworkers from SMAD4, separate this complex. That's what we call destabilize the complex. And for that, we are working with people that have small molecules, so that's the traditional drugs that everybody has, or peptides, which is in fact small chain of amino acids that are going to come in between and destabilize the complex. So those are two approaches that we chose to develop at the foundation so that we can have something, have the first idea of a treatment. Okay, so that's, now I would like you to have some questions maybe. I'm going to stop sharing. Where is it? Where are you? You're here. Okay. Oh. Okay, so questions?
We can cover questions about what Amel has just talked about, or as I mentioned at the beginning, anything else, anything that's been on your mind about research that we're not covering today that you have questions about. It's really over to you guys on what you'd like to hear about. Victoria, so you're unmuted.
Yes. So I have a question about, I think I understand the interruption of or targeting the SMAD4 and the SMAD2/3, like that's where a drug is going to target, right?
Yeah.
Or interrupt, right? Okay. Is there a specific physical characteristic or a physical, like, I know a lot of the individuals that have Myhre's have heart conditions, or they have, is there a specific physical thing that is targeted in that, or is it a broad spectrum?
So that the idea is,
does that make sense?
Yeah, I understand. So the idea is that this interaction happens in every cell, in the heart, in the brain, in the kidney, in the liver, in the immune system. Everything is based on this interaction, or interaction I simplified just SMAD2/3, but SMAD4 interacts also with other the same way. So SMAD4 forms complexes with other in every cells. So the idea is that we are not going to look only at your heart or only at your brain. We are going to work everywhere in your body, immune system, liver, lungs, gastrointestinal, brain, how to destabilize these complexes so that when we give a drug, it's going to act on everything, not just on the heart or just on the brain, but really everything, everywhere in your body. Because the thing is that SMAD4 is so important for so many different physiological functions, from the heart, the brain, the liver, the kidney, is that we are not going to focus on one thing. We are going to focus on SMAD4 but everywhere in your body. So the drug is going to act on SMAD4, but not only in one organ, in every organs.
Makes sense. Thanks.
So that's the reason why we need biologists and chemists that are going to focus just on SMAD4. And then when we have a good molecules, a good new drug, then we test it in organoids, so organs. Then we test it in whole organisms, so animal models, mice. And if everything is okay, if the toxicity is low, then we give it to the physicians and the clinicians, and they are going to test it with the agreement of the FDA or the EMA in Europe. They are going to test it on patients. But first, we need to start from the beginning, which is at the cellular level, even at the molecular level. Not thinking about the heart or anything, just find the right drug that we will be able to give to the whole body so that we can act on everything at the same time.
Thanks.
I have a question. And it may not be able to be answered, but it's okay. Like, is there like times and do you know if there's periods where like the SMAD4 is like I don't know how to like less active or more active? And you know what I'm saying? Like, and I think about like, I mean, obviously I'm 43 or whatever, but like there are times when like my health issues are amplified and more active and more things are added. And then there are times when like it's almost like it doesn't exist. You know what I'm saying? I mean, not that that's a really bad analogy, but that's the only way I can think of it. So I'm just curious if we know enough research to know like if there are times when the SMAD4 and is that something that is due to age or something? Because obviously they've not had, you know, like a lot of research on people in my demographic or whatever.
Yeah. So we know that SMAD4 is more active during development in embryology. So before you were born, and for the first 2 years, for the central nervous system, SMAD4 is really very, very important to decide which cell is going to become a neuron and which cell, sorry, is going to become an astrocyte. So it's very important during that time. But then after, if you look at what SMAD4, what coworkers SMAD4 is going to activate, you can see that there is, it's really involved in everything that is proliferation of the cells. So if you are getting older, the proliferation of your cells are going to be modified. The rate is modified, and you have also the hormonal aspect that interferes. So so that can be the SMAD4 function can be modified by your age, but it can be also modified by inflammation. So if you have inflammation, it can be modified if you have, since it's acting on your immune system also, on your T cells, which is the lymphocyte T. If you have infection, then the SMAD4 is going to behave differently and have a different function than you don't have an infection. So that's it's a very, very
You're saying, and
question.
correct. I think that's what I'm experiencing. You know what I'm saying? So like if you like were to like look like historically or biologically or whatever, the natural history or whatever it's called, you know, like I mean, again, I mean, obviously I wasn't diagnosed until my 30s or until 32, 33, you know, and people often wonder, how is that even possible? I mean, of course it's possible. I was born in the 80s and they don't do genetic testing like they do now, you know what I'm saying? So somehow I just got snuck under the rug, you know, kind of thing. But if you think about like in my childhood, I was a very sick child, just like everybody else. And then somebody along the way, between like around the age of 18 or so until mid 20s, maybe mid to late 20s, other than just like hearing loss and being diabetic, no real issues. But um, it went all downhill from there. So, and it's just, so that kind of like explains, that process, what exactly what you're saying biologically. I'm saying, humanistically or whatever, this is what they're definitely on the same like page or line, and until recently, until we were seen by the people at NIH, doing the immune study, I had never actually really seen an immune doctor, and they really explained like the T cells and all those things and they're tracking it really well at Mayo and NIH and stuff. So it's been really fascinating, that kind of thing. So I appreciate you sharing and talking about and being able to answer what you could about like the lifespan or kind of thing.
Yeah, that's really the everybody's going to react differently because of the what you live in your life. You have aggression of your body from infection, from scarring, and for example, one of the issue with SMAD4 is that you are going to trigger an over scarring tissue. Because you are going to proliferate more the cells that make the scars. So if you have any aggression, you have scarring everywhere in your body every day. Everybody has that. But with Myhre, you are going to develop over scarring tissue. So that's one of the issue, and maybe one person who would have less aggression of their body will have less reaction from the SMAD4. So everybody will be different.
And I'm assuming that could be different with the variants as well, right? The different variants.
Yeah, there's a small difference. Not compared to other diseases, the disease is quite similar from one variant to another. You have other diseases where you have really totally different phenotypes from one variant to another. Here you have just a small difference, but mainly everybody goes through the same issues, more or less.
Correct. Thank you.
Thanks, Holly. That's really sparked a thought in my head that I've been, and forget that I'm now ED of MSF. I'm now thinking of as a parent. So this inflammation concept has really fascinated me about like diseases in general and rare diseases. And Amel, can you, if you can, explain more about the role inflammation plays, and if we fix what we want to fix, that has a direct impact on inflammation? Or because when I've spoken to people about inflammation in the past, it can be such a hard thing to track because of the amount of factors and pinpointing if inflammation is increased because of Myhre, or if it's like that kind of thing. So can you speak to inflammation a little bit more and how that might be cured or helped or reduced?
Yeah, the thing is that so Myhre, the SMAD4 is at the end of a signal, as you said, from the external of the cells that is going through. One of the main one is TGF-beta, and TGF-beta is a cytokine. So you heard about cytokines during COVID, for example, saying that you had a cytokine storm. So it's a kind of signal that goes to the cell and that is involved in many other mechanisms that are in the inflammation. So it's not that it's really inflammation, it's one part of other mechanisms that are part of the inflammation. And in this inflammation process that is very complicated and large, one of the mechanisms is the proliferation of the fibroblasts that will trigger the fibrosis, and the modification of a type of cells just for you to know, it's epithelial cells that are going to be transformed in other type of cells that are myofibroblasts and that can contract. So it's a process that we call EMT in biology, and the SMAD4 is involved in this process. And when you have this process that is done, at the end you will have the fibroblast instead of the epithelium. So the epithelium is like that forms, they are cubic cells, that like Lego blocks, and they can make walls. Okay, and those cubic cells are going to become like stellar, like stars in the sky, instead of being cubic. So you imagine that the function is going, it's not the same function at all. So that's what's happening when you do a scar. You are going to cut this epithelial. So those epithelial are going to be transformed in fibroblasts. They are going to reproduce and rebuild at the end this kind of wall. But you have, so that's the reason why when you have a scar, I have one here, I don't know if you can, well, you can see it. If you have a scar, it's different from the rest of the tissue because it's different cells and the whole structure is different. And SMAD4 is involved in that, and you will have too many fibroblasts. And those fibroblasts are going to secrete, what do I say, proteins that are really built and strong, that are called collagen, fibronectin. So it's the extracellular matrix that is going to be produced too much because SMAD4 switch on genes too long, and those genes are going to be expressed in too big amounts. So you will have all this extracellular matrix that is going to be very strong, and instead of having something that is flexible, there are so many bricks instead of having just, I don't know, you're good in analogy, Kate. So the whole wall, instead of being flexible, is going to be stronger and stronger and stronger. And that's when you get issues, like if you need, in your joints, it needs to be flexible. So if it becomes too strong, then you have issues. If you have scar tissues that are really strong, then around your heart, around the walls of your arteries need to be flexible. If you become stiff and bigger, then you have stenosis. So there is a lot of this process that is the scarring process, that it's part of the process of inflammation, triggers that also. Then you have all the issues with Myhre. And there are new papers coming out that Myhre is also involved in the T cells activations. So it's all around inflammation. It's not the same as the inflammation when you have a bacterial infection, for example. It's not exactly the same processes, but it's similar to it.
Okay. No, that makes sense. And I guess that really ties back into the fact that rather than treating the component parts, that's why you need to go to the root cause, because the process is coming from the root cause.
Yeah, and it's coming to different cells, the T cells, the fibroblasts, the epithelial cells. So if you try to treat only one part, then you won't get a full results. You really need to treat from the beginning of the cause, which is either the shape or the mistake in the DNA. So something else that I'm really now actively working on is find other molecules that can act not only on the SMAD4 link with others, but on all this process of transformation of this EMT process that I was referring. So the epithelial cubic cells that become myofibroblasts. So I'm talking to companies that have some of those molecules to see if we can adapt that to Myhre syndrome. Is there any other questions? All clear. Is there anything you want me to talk about next time? I wanted to talk about fibrosis next time. Are you interested in that? Understand fibrosis, stenosis, the mechanisms, and how we can tackle this aspect?
I'm very interested.
Yeah, I'm interested. Absolutely.
Okay. Okay, so Kate, you take note.
Yes,
That
yeah.
will be the next one. Explain a little bit all this EMT fibrosis and the way we can approach that for Myhre, especially.
That sounds good. And if between now and the next one, I'll send you all out an email today with all the rest of the dates, just in case you hadn't caught them, so that you can put them in your calendar straight away. And I'll always send the link just a day or two ahead, so that it prompts you as a reminder. But if between now and then you think of an idea or think of something we haven't covered, please just drop me an email. We're very happy to be extremely flexible. Mel can answer questions on the fly. As we know, she's very experienced, and we can talk about anything you like. So yeah, just let us know what you'd like to cover as well as we move forward. Okay, any final thoughts before we let you continue your Saturday? No? Well, thank you. Oh, Nancy?
Yeah, I just want to say thank you. I really appreciate your efforts in discussion today. It's great.
Well, thank you for being here. That's very important. The more I hear about your feelings and not feelings, your questions and what you're worried about and what you don't understand, the better we will do our job. All of us. Sorry, that's my dog coming in saying hello.
I like seeing all the pets. We've got a pet. You had my cat earlier. You've got some dogs here with Holly. Bring your pet to the Q&A.
Hey, I'd just like to remind people, if you have any questions about donating samples or participating in the patient registry, just to reach out to one of us, and we're happy to walk you through it.
Yeah, absolutely. And Holly, we'll get back to you on the port question as well. I've already sent that off for that.
Perfect. That you said the patient registry, is that the one that we are always use and we update every year? That's not a different one, right?
Yes, no, it's same one from Cords, facilitated by Cords.
Perfect. I just wanted to make sure it wasn't like missing something. I mean, we're in back to school mode as a teacher, and I might be missing something. And so I wanted to make sure that I wasn't missing it. I know I haven't updated it yet for this year, so I will work on that hopefully this weekend.
Amazing, that would be great. And you might notice there's some new questions, not too many, because we try and obviously just try and keep it tight for you. Don't want to have you there too long, but there's some new questions as well that talk about mental health, that talk about caregiver burden, and it's just some other things that we feel important to understand about your journey and your family's journey.
Awesome. Sounds good. Thank you.
Great. Okay, lovely.
Great. And we are going to modify these capital letters and small letters for the gene versus the protein done. That was a really good remark. We need to be accurate.
I was just trying to help people as they dive into literature on their own.
Yeah. Thank you.
Yeah, thank you, everyone. Lovely. Okay, I will get a recording up of this in the week. Thank you so much for being here. Look forward to seeing you again next month. I popped the video into the chat, so please have a view of it, share it with your friends and family. Hopefully it's a great way for them to understand a bit more